I am an ambassador for the COP for Ghana, and today we have two speakers also from Ghana to share with us lessons and their experiences on data organization. Our first speaker is Mr. Samuel Kwesi Benefo, who is an assistant research scientist at the Council for Scientific and Industrial Research Soil Research Institute in Ghana. And our second speaker is going to be Mr. Alexander OWUSU ANSAH, who is a GIS analyst and also a soil scientist at the CSIR-Soil Research Institute of Ghana. Before I hand over the microphone to Mr. Benefo to present, I would want to make a short presentation about the COP to all of us. So kindly give me some few minutes to do this. I would share my screen. Okay. There you go. Share.
Can see it, so the owner can see it also.
Can you see my screen?
Yeah.
You can see it?
Yeah.
I have to change the settings here to swap. Please, can you see my screen? I hope everybody can see my screen.
Yeah.
Okay, so the COP is one of the components of the National Soil Information Systems Program coordinated by ISRIC. And it is a community of practice of soil experts who are on this platform to share knowledge and also educate ourselves on soil resources and data usage. The COP has soil information user considerations, which forms the basic workflow. And you can see that on the right-hand side from data collection, laboratory analysis, soil archiving, data organization, modeling and mapping, and applying soil information, and also data and info serving. The other component of the Soil Information Systems Program is the Soil Resources Library for soil information producers. The background of the COP started in May 2023 with 126 members currently, and we started with 126 members, and now we have 700 members within the COP from 42 African countries. Currently, also, we have 16 ambassadors from 11 African countries. The vision of the COP is to share ideas and strengthen each other's work, also collectively guide discussions and professional development, and also innovate for soil information systems development and co-develop standards for all soil information workflow steps. The goals of the COP is 1, knowledge sharing, 2, to foster collaboration and networking, and 3, to educate and also to strengthen our capacity. Activities include showcase events, joint publications and reports, also trainings, and also regular interactions via communication and networking platforms. To become a member of the COP, you can scan the QR code to be a member. Also, you can follow and learn more about the community via the ISRIC website, and also follow on LinkedIn for more interaction activities. The second component of the National Soil Information Systems Program is the Resource Library, which is a package of tools, tutorials, books, guides, and other resources to help through the soil information workflow steps already outlined, from data collection through to data and info serving. To get access to the resource library, you can scan the QR code, or you can use the email, the address, the web address, that is resources.isric.org. Thank you for listening. And now I would hand over to Mr. Benefo to give us the presentation. Stop sharing.
Thank you very much. Please, am I loud and clear?
Yes,
Yes.
we can hear you. I have to stop
Thank you very
sharing.
much. So can I share? Good. I want to share my screen.
Okay.
Next. Share screen. Okay.
So as Mr. Benefo is trying to share his screen, he is going to present on introduction to data organization. He would make a presentation. He has 15 minutes to make the presentation, after which we would have 5 minutes to ask him questions. So you are entreated to write your questions down, and after his presentation, we'll give you the floor to ask your questions. After which, we will take the second presentation from Mr. Alexander OWUSU ANSAH, who would present on Ghana Soil Data Organization, an overview of soil data management and applications in Ghana. If you can share your screen now, Mr. Benefo, you can.
That's fine.
Yes.
Is that okay? I've shared my screen. Please, can you see my screen?
Not yet.
Not yet.
Yes.
Yes, we can see your screen now.
Thank you very much. Scientists and upcoming scientists, I'm privileged to have this platform to also share what I know or what I've been doing with this community. Thank you very much. I'm Samuel Benefo from Soil Research Institute, Ghana, and I'm touching on the introduction to data organization. This very presentation is targeted to beginners, those who love to be in the soil science field, and they want to know what we do. And so those of the experts here, I know we have a lot of experts here, so it will serve as a form of refreshing what we already know. So my presentation will go through introduction, then the types of data, then the data collection and entry, and conclusion. In science, or in soil science, we refer data organization as structured and systematic management of soil data collected from fieldwork, laboratory analysis, and research activities. Again, it's a method of classifying or arranging data to make meaningful impact for the scientific community. Again, it involves methods and tools to efficiently store, classify, and manage information to make it accessible and useful for analysis, reporting, and sharing. To me, data organization starts from the beginning of the research. Before you start your research, before you start your trial, data organization starts. Because before you set up, you think of your objective, you set your objectives. Before your objectives are set, it means that there's data organization working. What are you going to pick? What kind of parameters are you going to pick? How are you going to pick them? How are you going to arrange them in the field? And so most of the time we focus more on picking the values we get, as then we organize it and we present to the scientific community. But we don't normally take cue from the field, or what we take from the field, which is very, very important. And so this very presentation will touch more on how we arrange ourselves, we organize data collection at the field. Now, with this data, we are talking about a bit of what we as soil scientists, we go through. We check the physical data, this is the physical chemical properties of soils, and that's the soil's texture, bulk density, structure, porosity, and yeah. Then we talk about the chemical data, that is pH level, organic matter, nutrient levels, metals, and what have you. Then we also deal with biological data, the soil microbial activity, soil fauna, fungal, and bacterial growth studies, and biomass and distribution. Then, as a soil scientist too, we also organize our data through getting some information on the hydrological studies or hydrological data, which is the water holding capacity, infiltration rate, hydraulic conductivity, and what have you. Before you set up your experiment, these parameters come into mind. And so depending on how your objectives are, you arrange how you pick the soil. Probably you just pick the soil sample and put them in a sampling box and go away. But there are a lot of other things that you need to pick so that when you get back to the laboratory, you may use those data that you might think it was not necessary. Now, in this picture, we have some of these data that we pick. As the left one, that's the soil infiltration, then bulk density in the middle, then a normal soil sampling for physicochemical properties. Then we get into the biological side, where fungal and bacterial studies, microbial studies also go on. So these are some of the parameters or exercises that we pick from the field before we come back to the field. Now, we look at field data organization. What do we do when we get to the field? The most important organization of the data is by taking care of or picking the GPS coordinates of these of the sampling area. If you don't pick them and you leave, you cannot come back and replicate your study. And so it is important to pick the GPS coordinates. Then we also look at some of the sampling depth. Why do we pick the sampling depth? From 0 to 15 or 15 to 30, you decide why are you picking that depth. And it forms a major part of organizing your data right from the field. Because if you pick it wrongly, you get bad results that you may not need. And so if you are looking at why you pick the soil depth at maybe 0 to 50, it means that you are looking for information beyond 30 centimeters down into the soil. And so that's depending on your objectives, you look at that. Now we look at the texture, the moisture, and the environmental conditions. I will later touch on the environmental conditions in this presentation. Then we look at laboratory data management. And this is the document data that we take from the field. We analyze them at the laboratory. You know, when it goes through the laboratory processes, we put them together. Even though we put them together, because the sample bags are labeled, you are able to really organize them. If it is about NPK, if it's about pH, if it's about soil moisture, if it's about organic matter that you are looking for, after going through all these processes in the lab, you arrange them very well on the spreadsheet. This is where the scientific community will be interested, because you have to use that to communicate to anyone who was not part of this of the project or of the study, that this is what you did and this is what you have done. And now if the person look at your data sheet, he should be able to even continue by analyzing the data for you into the software using your objectives. And so if you don't organize your data very well on this spreadsheet, it will be very, very difficult for any other person, an independent body, to take your data and work with it. And as scientists, we know that the data is our Bible, and that once we are able to organize it very well and present it to another person to study or do analysis, it is easier said than done. But if it's haphazardly arranged on the sheet, it doesn't conform to what you want. The sampling depth, it doesn't conform to it. Everything has been scattered. It becomes very, very difficult. So one major problem that some people who are upcoming, most of them, they don't really know how to rearrange the parameters that they have really gone through the laboratory. They just place them, and at the end of the day, they are unable to analyze. And some of these softwares like the R and the Python, you really need to arrange it very well to conform to the right shape before it will be able to analyze for you. So this is about laboratory management. Now, this is what I was talking about. This is a bit of a picture of work that I've done already. You know, we are looking at the leaf chlorophyll content. We are looking at the stem girth, the height, the leaf length, and the leaf width. You see how it has been arranged. This is proper organization of data. And that once I am through with this, I can leave and give my data to another scientist, use the objectives, and just do the analysis for me. And so we have to go through this process, make it very neat and concise, precise, then we are able to communicate very well to the scientific community. Now we are looking at the classifications and standardization of this data we are talking about. And it is very, very, very important that we take a cue from the World Reference-Based Soil Resources, because that is the hub of soil. It contains all the categories of soils, the classifications, the qualities, and whatever we are to see in particular type of soil, it has been grouped and described. And so we look at you, after getting your data, you have to peg it to the World Reference Soil Base RB, WRB for short, to make sure you are able to classify the soils that you are working with. And that is very important because the fertility status of a particular soil, soil type or soil series will not be the same for a different soil series. The way you attack or the way you manage a particular soil series may not be the same as another soil series. Some are loose, some are hard, some are crumb structure, you know, and so you really need to go through this and make sure that the categories conform to what you have gotten from the field before you are able to say, okay, my soil belongs to this type of soil and that I have to work it out from there. Then the next thing, very important, is using units. The units, making sure that you place unit by these data that you have arranged. Data without units is not data. That is what I always say. It is like I'm reading a storybook because I don't understand the figures you are giving me. So we should not forget making sure that as upcoming scientists, in quotes, since this presentation is geared, you know, much focused to these upcoming scientists or beginners, you have to really make sure that you include the unit to these figures so that you are able to analyze securely, make sure that your analysis is accurate and you come out with the best result that you desire. Now, this world is turning to, everything is turning to GIS system, it's data IT system. So we have to also have to peg ourselves to the GIS system. You know, using the GI, I'm not going into it because Mr. Alexander is already there and he's an expert in the GIS system. But what I know is that he's able to prepare soil maps for you, the topography, the spatial and the patterns and the land use, is able to arrange and draw it nicely for you. So wherever you pick your soil, this soil series, when you have a soil map is, what I know is it contains different soil series, and GIS is able to differentiate between these soil types that are in the particular area that you are working. So we really need to also imbibe GIS technology into the data organization, and to really, really help us. Yes, coordinating system and mapping, that is what I have already mentioned. It is very, very important that GIS inclusion into our work, add more bite to our output. Now we look at metadata documentation. Metadata, I mentioned that I will talk about the background information of soil area. It is the background information about the data that you are collecting. When, how, by whom, how was it collected? You see, if you are able to get that background data, more especially the soil condition to which we pick the soil. Let's say you work on bulk density. That is the mass per unit volume of soil. If you are working on bulk density, you take your call and pick your bulk density. What was the condition of the soil environment there before you pick the soil bulk density? Was it waterlogged? Was it that dry soil? Was it a bit moist? Whatever you get there will also be inclusive in your interpretation of your data. Because if I get to a field that is so moist and so soggy, you should know that how the soil will behave. But if I get to a hard ground, let's say particularly the northern side of the country, Ghana, at this year, especially in the lean season, very, very dry soil. Even if you are picking the soil bulk density, it's even difficult to use the core to pick it. And so you really have to tell the story about the background, how the soil environment was before you were picking that data. And that is proper organization. Because at the end of the day, after analysis, what else? It will tell you, okay, but why you are supposed to do this? In science, you are supposed to get this. You are supposed to get this. But your data is telling us another thing. So why? That is where the soil conditions or the environmental factors will come in and buttress your point. Now, we look at data storage and backup. We all know that we really have to store our data very well and make sure that easily we can retrieve it from any system that we place, and that we have to properly name our data files. We easily forget because of we are tired after working, we just close it up. If you need a data to work on a certain parameter or to work on another thing, it becomes very, very difficult for you to retrieve it. So proper naming or filing of data is very, very important. Then the next thing is backup strategies. And that is where we can use the external drives to, like a hub, take all your data, you put them in. Whenever you need it, you are able to get it. Now, in conclusion, I will say that effective data organization in soil science rely on combination of so many factors, the practices and technology tools, and ensuring that data is consistent. And that once we are able to, as soil scientists, to help others who are coming up to build their confidence in soil science, we must also tell them the nitty-gritties of how the soil behaves. We must expose the soil to them and also help them to improve their capacities in the soil environment. Thank you very much.
Thank you, Mr. Samuel Benefo, for your interesting presentation on Introduction to Data Organization. That was really insightful. We would have some few minutes for questions. So online participants, if you have any question, the floor is yours to ask our presenter any question you have. You can open your microphone and then ask the question.
You can also use the chat if you like to post your question. So far, I don't see any question, but until we receive any question from the audience, shall I start I start?
Okay, yeah, cool. You can start.
First of all, Dr. Samuel, thank you so much for joining us today. It's a pleasure to having you as a keynote speaker in our last showcase for CoP for this year. And my question is, do you have experience in data organization outside Ghana, or you only worked in Ghana? And if you do you see any similarity or differences in the challenges, the approach that other people use?
All right. Thank you very much for the question. Yes, I'm based in Ghana, and so far on the field, I've only been working in Ghanaian soils. But because I read a lot and I read papers and I follow scientists, I'm able to also know what they do and what they do differently and what to pick and what to also add. And so that is about it. So the experience is basically in Ghana. But I also have some friends, more especially in the Kenya area. Sometimes the way they pick their soil samples is different. You know, for here we use the auger. Yes, we use the auger to pick soil samples, but sometimes they also have their own calibrated auger, which is not what we use here. Then they hit it into they smash into the soils and they pick it in that shape, and they also take it as soil samples. You know, even in Ghana, some of the projects that we involve ourselves in, I may not mention any project, but some project come specifically protocols that we must use to pick soil samples, even though we know how to pick. But you know, people do things differently, and that you really have to conform to what your client needs so that you can satisfy them. Thank you.
And how, to what extent, following like different protocols or using different tools can affect the results of the data later in the lab? Or, I mean, of course there will be differences and effect, but to what extent you think? Like the example you gave from Kenya, what is kind of like a bit different than the one in Ghana. So using this massive different in tools for data collection and organization.
Okay.
To what extent that? Yeah.
Yes. There is an effect. Yes. Scientifically, there's an effect, especially when you are picking soil samples at different depths, maybe 2 depths, right? But after picking the first, they're using the auger because you pick it round, you know, in a clockwise manner as you drive it into the soil. When you remove it, you should be able to clear the outside of the auger, making sure that all debris and any other material or soil that is not part of what is inside will not go into your sampling bag. And more especially when you are getting to the second layer, which is maybe there's layer B or maybe the subsoil. If you still pass through the topsoil before you get to the subsoil, and that definitely there will be some soil particles from the topsoil adding up to the subsoil. And that is where cleaning of the auger is very important before you put them in the sampling bag. But if it's not done that way, and there is sort of fabrication, you just pick it, smash it, drive it, and pick your soil samples, the likelihood that soils from the A horizon, sort of in quote, may also join soils from the B horizon, where when you are at the laboratory and you want to do your physicochemical properties, you want to see it very well, you may not get the best result, but you might think it's the best result. And so we are all learning. I mean, I know that since this platform is great, I mean, it's fabulous. And we are learning a lot from this platform. I must tell you, I've learned a lot through the webinars and papers people are putting in. It is shaping us. So I'm thinking of maybe sometime soon, there will be a global protocol for certain kind of parameters or soil sampling so that we can.
I was going to ask you actually, like, because with the GSP, for example, they publish and people try to do the standardized method for lab, for data collection, data organization, but you think this is not shaped enough yet, or is it there but people not aware of, or they don't really follow it? And sorry that we see the hand of Dr. Burton, but this will be my last question, and then we will answer to Dr. Burton. Yeah.
Yes, it's true.
Did you get
yes.
Yeah.
Yes, I got it. I got it.
Yeah.
Yes. Yes. The announcement or for people are not really following. Most people are not really following the updated ways of picking soil samples or working with the soils. Some are using the old ways of what they do, and they are conversant. They are always okay with what they do. But some are also picking up and changing and upgrading themselves. So I think that by and by, as we move on, sometime soon, that is what I will say. I know by sometime soon we may have global units where a soil community, when we say A, we all know how to achieve A. When we say B, we all know what we go through to achieve B. And that is where I think when we get there, that will be perfect. Anyway, thank you.
Yeah, thanks a lot. Dr. Burton, would you like to ask your question?
Yes, thank you very much. And Eric, good evening. Good evening to you.
Good evening. Very lovely to hear from you. It's been a while. Thanks for joining.
It's been a while, eh? Yeah.
Yeah.
We have been on this side very busy too, moving from one field to another one. Yeah.
I can imagine. Yeah.
It's not been
Thanks
easy.
for joining. Thanks so much. Please ask your question, Dr. Burton.
Yeah, I want to first thank Samuel for the presentation. That he really gave an overview of the process, and I think it's like a summary of the various steps that is being used to implement and get the final product. So I'm really very happy with that, and I think such type of presentation could be developed in a small guideline of manual, something like that for people that are really new into the process. Sammy, I want to find out from you. Yeah, actually, my question focuses more on source sample analysis because it is actually a very big challenge for most of the initiatives in Africa because people really can be able to struggle and get the source sample very well from the field, following the good protocols. But at the same time, to analyze and get the information or the soil property needed is really a challenge. First, because of the cost involved, the quality of the laboratory available, and other challenge in Ghana that you have like source sample well collected, well processed. At the same time, you are really not able to get the satisfactory result from the laboratory because either the cost is too high or the results sometimes are not really very accurate or up to the expectations. And the last aspect is, are you people already involved there in soil spectroscopy to like support the wet chemistry analysis as a way to reduce cost and other kind of thing? That is just my point. I wanted us to see if you have some other experience in that, particularly from Ghana. Thank you.
Thank you. Thank you very much, Dr. Burton. Burton, thank you so much. Yes, for your first question, it is really a worry, because of financial implications or the cost of analyzing soil samples. Most times, students in particular, they may have to reduce the sample size, even though they pick a lot of parameters, they pick a lot of soil samples that will at least speak to the project or speak well to or help him build more results through the project. But unfortunately, because of financial implications, they are bent on picking maybe few soil samples, then they will come to the lab. Even then, they will also plead for reduction. And that when the data is out, you can see that even though it's not hollow, but the data is not heavy. Normally, I use the word heavy because if you have a large sample size, then you are confident you can speak to the results that you have gotten. And that is a challenge. And we are looking for a way that donor or agencies or organizations will really delve into this, especially for students who are learning, so they can really help them through their laboratory analysis. If you don't go through laboratory, especially in the field work, I mean, you cannot graduate. You cannot get any meaningful results. And so it's also happening in Ghana. Yes, I'm at the Soil Research Institute, Ghana, and I'm glad to tell you that our laboratory is certified. It's a certified laboratory that most of the parameters are being done. But the cost, as you mentioned, because they buy chemicals. And so the financial situation is really a worry. But for the spectroscopy, I am not too sure because I'm not at the lab and I don't really know the details much of what they do currently. So I may have to find out then, since we are in one community, I will come back and present on that and see how best we can handle it. Thank you very much, Dr. Burton.
Thank you, Mr. Benefo, for the answers. There is one question in the chat. Maybe we can take that quickly. And then briefly, Mr. Alex would also have the opportunity to say his question. So in the chat, we have a question from Dr. Irene Bame, and she's asking, in some cases, topsoil is collected from 0 to 20 centimeters, and in others, it is 0 to 30 centimeters. It is possible to harmonize the data collected from these different depths from different locations. So yeah, it's rather a question. Is it possible to harmonize the data collected from these different depths from different locations? That is the question. I hope it's clear.
No, it's not that clear, please. Can you come again, please? I'm getting some hitches in the internet. Please, can you come back?
So the question is in the chat, and Dr. Irene is asking that in some cases, topsoils is collected from 0 to 20 centimeters and some from 0 to 30 centimeters. And she wants to know that, is it possible to harmonize the data collected from these locations or from these different depths from these different locations?
For topsoil, so a lot of activities goes on in the topsoil, even from 0 to 10 centimeters. That is where a lot of these nutrients they reside. And per literature and what we have read, if we go much deeper, it means that there is loss of nutrients. Nutrient availability lessens as much as you go much deeper. And so if you are working on maybe the area is for shallow rooted crops, I mean, it is always good to make sure that you sample between 0 to 15. I mean, you can harmonize the 0 to 15 so that you can really know what the soil is, the nutrients levels of the soil, the condition, everything is so that you can really match up with your recommendations to the farmer or whoever is tasking you on that. And so for the different depths, yes, people pick it at different depths, but for my level and for the experience that I have gotten with data that we have analyzed in the laboratory, if you focus more at the top, you get more of the results accurate. But once you get much deeper, you mix up and you may not get the right or the right nutrient levels that you need in the topsoil.
Okay, thank you, Mr. Benefo. I think the discussion is becoming very interesting, and we have one more presentation to go. So if you would agree with me, I would invite Mr. Alexander OWUSU ANSAH to make his presentation. And after his presentations, we can come back to all the remaining questions.
Hello, Eric.
Hello, Alex.
Yeah, I wanted to add up to a question asked on the spectrometer.
Okay.
Yeah, soil research, we do that, and it depends on the quantity of samples in the lab. So if there are a lot of samples, over 1,000, then we have to put some into MRL to determine its parameter, and others a percentage goes into the wet chemistry to predict the other parameters for them. So we do that in SRI, soil research. So I wanted to make it clear to also bring down costs.
Okay, thank you, Alex, for clarifying that for us. Now you have the floor to also present, and Mr. Alex is going to present on Ghana Soil Data Organization, an overview of the soil data management and applications in Ghana. Please, Mr. Alex, the floor is yours.
Okay, please, can you hear me?
Yes, we can hear you.
Yes, Alexander, we can hear you, but we cannot see your slides.
Yeah, I'm trying to, I'm using a tablet, so I'm trying to load it from a tablet.
Okay, thank you.
Please, can you see it?
Yes, we can see it. Can you? Yes, thank you. You can take it over.
Thank you very much. I am Alexander OWUSU ANSAH from Soil Research Institute, Ghana, and I'm presenting on the Ghana Soil Data Organization, an overview of soil data management and applications in Ghana. First of all, in Ghana, if you say institutions that organize soil data, we have the Council for Scientific and Industrial Research, Soil Research Institute, the Ministry of Food and Agriculture, and then the universities. And what we do is, it involves data collection and then storage, and also the data, we don't just go and collect them, but for us to delve into them and then apply them to improve on agriculture, land management, and also policy making. So we have the institute involved. We have the Soil Research Institute, which is the main body when it comes to soil research. And we have the Ministry of Food and Agriculture. We have the universities. That's when it comes to the soil department in the universities, and also private agronomy firms, like the international donors and funders, fertilizer companies. They are all part of entities that helps in collection of data, and also when it comes to information on soils in the country. So data collection methods. Every data collection has its own objective and what to achieve. So we have the field surveys, mostly which we use in soil mapping. And also we collect data from published papers, and also technical reports also. Then even in maps, the GPS mapping, like data collected from the field, the GPS coordinates and all other information you get from that. That's also another way of collecting soil data. And then also when it comes to the lab, all soil samples coming to the lab is a way of collecting information on the soils. So all parameters analyzed and every aspect of the soil analyzed in the lab, all these informations are being collected and then being processed. What do we do with all this information that we get? That is the data management. We have to store them in a system that we don't have to compromise losing them. So we have the servers, and also we create databases and host them on servers and also in the clouds. And we are managing them. So we have to be able to use this data for decision making. So we analyze this data and then we make decision out of this data, and then also ensures easy access and sharing. Mostly our collected data and collected data, we host, or FAO hosts trainings, and then we share this information to generate maps when it comes to the continent. So we do that to ensure access and also sharing of data. Role of GIS and remote sensing, we mostly use GIS and remote sensing tools. So we also use our statistical tool in predicting and also modeling soil properties. We also use the Google Earth Engine and also USGS Earth Explorer when it comes to land use and also climatic data. We use these tools to also derive information from there. And also we have the ArcGIS Pro, the QGIS, and other GIS packages that we also use when it comes to data organizations. All this data we collect is to inform or advise, or as an advisory services. So the soil data helps to create or design crop selection and planting. And mostly when it comes to crop selection, we talk of the soil suitability. What are the soils suitable for? Most of this data is to advise people who wants to go into agriculture what land acquired can be used for when it comes to the soil. So the crop suitability, crop selection, we advise on them, the planting. And also we have the fertilizer use irrigation. There's analysis done in the lab. We base on this information and also generates fertilizer recommendations, and also advise on irrigation when it comes to soil water conservation and how to also conserve water in the soil for plant use. And we also have the environmental management. This data helps especially in environmental assessment. We use this soil data to also help in managing the environment. And then soil data also helps researchers to design climate resilient agricultural options, and then also strategize to how to cope with soil changes. Especially, I'm currently in the northern part of the country, and it's dry, and then the ground is also dry. So if you want to plant anything by now, you don't have any means to do because there's no moisture in the soil. So it also helps, this information also helps to design strategies to how to manage the soil when it comes to climate issues. So that's what these data sets we are collecting or organizing from different parts of the country helps us to do. We have challenges as a country, limited funding, and then also outdated equipment. There may be some, but they are limited. So we have to improve on them. And then few people are also in the soil fraternity. So we need to also build capacity and broaden our horizon to build capacity. And we all know the world is changing with modern way of doing things. So we have to all be aligned the same way to be able to function well. So we need to build capacity in that way. And when it comes to data sharing, because there are a lot of entities or institutions taking data or collecting data sets, it makes it very difficult to bring all this information into one pool when it comes to organizing data. So that's the challenge as a country. It's very difficult to get all data collected across the country into a pool. So that's one challenge of our data organization. Data organization is also creating opportunities. One is using our machine learning to generate informations, and also the drones to give you time information. And then now we have the Internet of Things. You can sit behind your laptop or your phone and then request for information online. So we are moving to that era that we can use this data collected to do a lot of things. And it's also giving us an opportunity to also bring the National Soil Information System into function. So that's what this data organization that we've been working on over the years is bringing us to. As I said, some of these soil data collected, we use them in mapping. So the map here is a map produced with FAO, the soil organic carbon stocks of Ghana. So we could collaborate, share data, and also come up with maps. And then, as I said, these projects and data collection also helps us to identify suitable soils for crop farming, and also helps to bring up recommendation for fertilizer for improving crop yields. So we have future prospects in data organization, and that is when the National Soil Information System is functioning well. It will help us to consolidate all soil data and also enhance accessibility for stakeholders. So that's the future prospects we have now with the data organization. So in conclusion, I would say soil data organization is essential for sustainable agriculture, environmental management, and climate adaptation in Ghana. So we are to improve data management, and then this will lead to better land use and agriculture productivity. Thank you. Hello.
Hello, thank you, Mr. Alex, for the nice presentation on the overview of soil data organization in Ghana, its management and its applications. Thank you for the presentation. The floor is open for questions from our audience. So please, if you have any question, you can open your microphone and ask Mr. Alex. I'm also looking at the chat. If there is any, I would read it out.
We have a question I'm not sure if it's for Alex. I think it's for our first speaker, Dr. Samuel, right? It's from Adine. She's saying for irrigation project, which one is better? First option is a auger sample or 2 profile pot sample. Dr. Sam, are you here?
No, I think Dr. Samuel is not online. He sent a message that he has to leave to attend to another activity. So if there is anyone in the audience who would want to answer that, I think it will be interesting to know from the audience as well. Dr. Aden asked, Dr. Kome, please, you can open your microphone and answer to that. So Dr. Aden Omar was asking for irrigation project, which one is better, auger sample or soil profile pot sample? Dr. Kome, please, the floor.
Okay, thank you, Sam. Thank you. Can you hear me?
Yes, Dr. Kome, we can hear you.
Okay, thank you. Good afternoon. Good morning to everyone. That's quite an interesting question. I just want to share that it will depend on the objective that we have at hand. Usually there are some activities like greenhouse agriculture farming where we do grow crops in pots. So usually soil is transferred and you see that the soil is actually obtained from surface horizons. You see that there augering will be best.
Yeah.
But once you get into the field where you have particular crops that are in the open, especially perennials, I think there we can think of going right down to the soil profile. So I think it all depends on the type of activity we have at hand. That's my little contribution. Thank you.
Thank you, Dr. Kome, for the nice contribution. I agree with you. I think it depends on the objective. Your objective would guide you as to which method, whether to do the auger sample or profile. So I agree with you on that. Any more questions to Mr. Alex? We have one from Dr. Oluwaseun Olatunde. He says, great presentations. Greetings, everyone. The challenge of having a structural soil collection protocol for various regions, especially in Africa, is what ISRIC can work on. And how about getting grants to solve the challenge of stakeholders, that is, researchers, students, and farmers, having access to the soil labs to conduct soil analysis at subsidized rate? Yeah. So that is from Dr. Olatunde.
More like a remark. Yeah,
Yeah.
I think, thank you, Dr. Olatinuda, for your remark. And I think at ISRIC, we do have this capacity. You are correct for structuring a protocol for soil collection. But I think this is also depends on the objective again, because we do have these protocols, but it's still, we don't have one protocol that can be followed for different projects. It depends on the project's objective and what we need to do there. But I think it's possible if we have a protocol per purpose, let's say, or objective, then you are right. That's possible. And perhaps I will take this note to see how we can develop this further within the COP and with our colleagues in ISRIC, specifically Nils. He's an expert in the data organization. We also have Paul. So thank you very much for the remark.
Yeah.
And can I ask Alex to stop sharing, please? Alex, can you stop sharing the screen?
Okay.
Thank you so much.
Okay. Oh, how do I do it?
And can I also address the second remark? He's talking about the challenge, getting grants to solve the challenge of stakeholders having access to soil labs to conduct soil analysis. I don't think accessing to lab needs grants. I think it's more like you cannot really design such a proposal, but I believe that it's more about the national institutes where they should have a developed lab and make it open for stakeholders to use upon like agreement or perhaps simple fees that they can apply for maintaining the lab. But then that's also something more can be more arranged internally, I would say, on the regional level, for example, but also, yeah, so I think it's a nice idea to start from, but then you have to bring these stakeholders together and then see how you can manage this issue and then to find a funder that is interested into, because as you mentioned, the lab is it's really costly.
Yeah.
And maintaining lab and machine products are consuming products there for analysis is really not very attractive for donors, but it's rather like should be a strategy within the organization on how to maintain the lab and how to generate income that at least keeps the lab running for stakeholders. It's really complicated topic, yeah, to address, and I don't also see so much interest from donors to invest in lab. But yeah, thank you so much for the remark.
Yes, we have two hands. Dr.
Can we
Atinafu.
finish the chat first, Eric, if you
Okay.
allow me? So let's
Okay.
finish the chat first, and then we go to the audience to the. So we have most of time for irrigation. It is better doing profile sampling. Tell more detail than. Okay,
Yeah.
that's also a nice remark.
Yeah.
Yeah, thank you very much. Yeah, Eric.
Okay, thank you, Dr. Atinafu Tunebo La Febo. Please, you have the floor. I saw your hand up. So please, you can open your microphone and ask your question or comment.
Can we close the chat then?
Okay.
No, I have. Am I clear?
Yes,
Yes.
please, you are clear. We can hear you.
I try to respond on the chat spot. It is better to do for irrigation. Its profile sampling is more detailed than that of auger sampling. Auger sampling is very important for doing nutrient analysis on top soil. So irrigation related with deep soil. So we do profile sampling is.
Okay, thank you for the remark.
Yeah, thanks for the remark. Dr.
You're
Komi?
welcome.
Dr.
Komi, your hand is up. Yeah.
Okay, thank you once again. Thank you once again for giving me the floor. I first of all want to thank Alexander for his presentation. And something caught my attention. If I understood him so well, it seems soil data organization is a major challenge within the framework of organizing a national CIS. I would like to find out from Alexander what strategies or what measures are being taken to tackle this type of difficulty. Do they like have a database management system, or how do they tackle this type of problem? Because it is a big issue. Thank you.
Thank you, Dr. Comey. Mr. Alex, you would want to respond to Dr. Comey's question?
Thank you, Doc. It's true that there is a big challenge nationwide. The problem is most of these soil data collected are in the hands of institutions, and then I think currently or recently, SIX is trying, or with the help of district, stakeholder engagement has begun to bring together all entities that deal with soil information. So with time, they have to understand how the way we want to go, the approach we want to go for a win-win situation. And then I think maybe they will understand the need for sharing of data so that we can come up with a national data system. So we are in the process trying to do that. So in no time, I think we will get there. Stakeholders are now trying to understand the need to share, and then we are putting in place system that will check each one's data. That is, no one can use the other's data for anything but for the purpose of what CIS stands for. So we are on course. Thank you.
Yeah, thank you, Mr. Alex. To also add a little to what Alex have also explained, yeah, this is a big issue in terms of having a national database. So as Alex said, we started collaborating with ISRIC back in 2014 to have a harmonized database. So data come from different sources, reports from fieldwork, from the laboratory, and sometimes even data that comes to the laboratory, you don't have the coordinate assigned to them. You need to having secondary information in terms of the location, try to georeference the data to know the exact locations from master's thesis, from published articles. So developing a national CIS is a huge task, and it takes a lot of effort. So we adopted a publication by Johan Lenas, who is in ISRIC. He did a database, soil database management system that we followed to also adapt to Ghana. And as Alex said, it has been a challenge, but then still we keep on upgrading to fine-tune it, to be able to use it for various applications, for digital soil mapping, or soil fertility, fertilizer recommendations, and others. But then to get data from different sources, it takes a lot of effort, and gradually, I think we are getting there. Yeah, so that was the little I wanted to add to Mr. Alex. Yeah, thank you. Any more questions from our audience? I see Ashanafi. Ashanafi would also want to. Do you want to add something, because you have also been doing a lot in Ethiopia in terms of organizing data for Ethiopia? Today the focus is on Ghana, but it would be nice to also hear from Ethiopia, just to learn from Ethiopia as well.
Okay, thank you, Eric. Yeah, good evening and Good evening, everyone. Maybe it's 7 p.m. in Addis.
Yeah. Good evening. Good to hear from you, Ashinaf. It's been a while.
Yeah. Just so. Okay. Thank you for the opportunity. I think before the end of this year, we are planning to share our experience as well. So, as you mentioned, we collated, huge legacy data sets in Ethiopia. And then our experience, we started by, data ecosystem mapping, which institutions have, which data in what format. So we started, by data mapping. So, and then following that, we identified, institutions who are fully and partially willing to share data. So we formally request those institutions, and then we try to get data. But as you know, just the awareness and also the platform at every institutions, is very weak. And so we requested, formally, most of the data, were, in the hands of individuals, or if it is institution, in personal computers or in analog formats. So we quality, whatever in available and also accessible. And then after that, we started with a very simple template, not simple, but it's very, matured template developed by Johan Leras, as Eric mentioned, of that the template that is, developed for Africa Soil Profile Database. So we adapted that template, but after AFSIS, and there were 2 projects, and through those projects, we received also a very customized template through ASRIC with help of with a team support led by Johan. And then we use those templates, and we adapt it to our service, and we develop a kind of a very extended template that encompasses almost very comprehensive variables in all the likely available physical, chemical, and biological properties. And then we already populated those templates from the legacy data source. And then by now we collated that legacy soil profile datasets, which was organized by the Coalition of the Willing. And also there were one big project that conducted countrywide soil survey mission, which we normally call it ETHOSIS project. So through ETHOSIS, more than 70,000 topsoil datasets were collected. So that dataset was somehow in a structured format. So now we are working with ISRIC under enhancement, to implement ISO standard and standardize, whatever populated in a very comprehensive template. So now we are working to have a one data model, because as you know, legacy data is sourced and available in different formats, and also follow the different procedures. So now we are working to produce a kind of one data model following ISRIC ISO standard. And partially we started to produce some deliverables, and we are working also for some data sets to push to extraction and transform and loading process under the NISIS server. So, and then our aim is to have one the data model, which we do have a very harmonized and standardized legacy soil data sets that can be accessible to all national soil users. So this is the status that we are in. So at some point we'll share our experience through presentation, and hopefully before the end of this year. And I don't know if I think this session is maybe the final one, so we can discuss and arrange somewhere, because we are also supposed to finalize this enhancement project by January. So maybe so that we can demonstrate the intermediate products that we have been working on. Thank you.
Thank you, Ashinafi, for sharing these insightful experiences from Ethiopia to the group. I think it's been a good addition to our presenters today. Yeah. I think
No more questions. No?
Let me check the chat. Do we see any more questions in the chat?
No.
Okay. Yeah, then we thank our presenters once again. We thank them for sharing such nice experiences and wonderful presentations with us. It's been good having to hear from them. It's been a wonderful showcase. I would now invite Dr. Crow to say his final remarks before we close it for the day.
Yeah, thank you, Eric, and thanks for our both keynote speaker from Ghana for their presentation, and for all our audience for joining us today in this final showcase for the COP for this year. And thanks a lot for all your remarks, comments, and contribution. Thanks for your time, of course, and it was really great to have you and to join us in this event. For the next year, we will update all the COP member about the new strategy for our activity for coming year, 2025, and also expect a new announcement and link for our COP member to apply for being a COP leaders or ambassadors for 2025. Thanks once again, and wish you all a lovely afternoon and evening where you are. And we stay connected on the WhatsApp, hopefully. Yeah. Thanks, Eric.
Thank you, everyone, and have a wonderful evening wherever you are on the African continent. Thank you.
Thank you very much.
Thank you.
Thanks very much.
Thank you.
Bye.
A lot.
Bye-bye.
Thank you so much.
Thank you.
Thank you. Yeah.